Okay, so here we are looking at one of the specific aspects of redox chemistry that we need to look at for our course, and that is determining the amount of dissolved oxygen in water. And we do this via redox titration, known as the Winkler method. So let's have a look at it. So essentially, dissolved oxygen in water is looking at the amount of oxygen gas that actually dissolves from our atmosphere into our aquatic environment. And as you can imagine, this is pretty important because things like aquatic plants and animals, they still need oxygen. So they need oxygen for respiration. So if we don't have dissolved oxygen in water, or it's used up too quickly, it can cause problems with our ecosystem. So oxygen tends to enter the water from the surrounding air, but also from photosynthesis due to algae and bacteria and so forth through water sources. We want about 5 parts per million, which is equivalent to about 5 milligrams per litre, in order to maintain a healthy water environment. Some fish, such as trout, okay, which you usually find in rapids and things like that, require 8.3 ppm. So it changes the environment that we find these organisms depending on the amount of dissolved oxygen they require. Typically, when things fall below 3 ppm, the water becomes very polluted and it essentially becomes very difficult or impossible for aquatic life. So we consider that a high level of pollution. This diagram here shows some of the ways that dissolved oxygen can get into the water. So aeration over rocks and rapids, diffusion from the environment, or photosynthesis due to aquatic plants and things like that in the water system. Hopefully you remember that with temperature, gas solubility decreases. So the dissolved oxygen that we find in a waterway can vary depending on the temperature and weather of the day, but also of the season. Okay, this is directly related to that idea that the solubility of gases decreases with increasing temperature. So as you can expect, global warming, rising ocean temperatures are great factors for our dissolved oxygen. If our temperatures of our seas are increasing, then the amount of dissolved oxygen in those environments will decrease, and that will affect things such as corals and other plants. So biological oxygen demand is a little bit different to dissolved oxygen. It is linked in many ways, but essentially it's the amount of dissolved oxygen needed by aerobic decomposers, so decomposers in our waterways, to break down the organic material. So there's lots of organic materials, fish waste, dead plants, and various other things in waterways, and if there isn't enough oxygen present in that, there isn't enough, we don't have the facility for aerobic decomposition. The cleaner the water, the lower the biochemical oxygen demand, okay, because there's less things living in it, there's less stuff having to be decomposed and various other things. So that's around 1 ppm. But water that is considered to be polluted has much higher levels, around 5 ppm. Okay, so if we look here, we can see different effects from industry as well. In terms of the effluent or waste from a brewery, we'll have around 500. Water from an abattoir has around 3,000, and that makes sense because of the large amount of organic waste that would be associated with meat processing. So we're looking at this, we need to kind of know the difference between aerobic and anaerobic decomposition. Most of you do biology, so this should be fairly straightforward. Aerobic is the most common form of decomposition, and we see that it uses oxygen and it oxidizes other elements. So you can see here that the carbon here is being oxidized by oxygen, forming CO2. In the case of anaerobic decomposition, of course, there's no oxygen around, which means that in this case, our organic components are actually reduced. So they combine with hydrogen, forming things like methane. So we tend to see this anaerobic decomposition happening in landfill, also in sewage treatment and things like that, where it's not highly aerated and we don't have access to the oxygen. So if we look at carbon, under aerobic conditions it will produce CO2. Under anaerobic conditions it will form methane. Okay, hydrogen will form water, but in anaerobic conditions it can form a number of different things, including methane, ammonia, hydrogen sulfide, and things like that, which tend to all have bad odors and are associated with rotting organic matter. Nitrogen will be oxidized in the presence of oxygen to form nitrates, and then we get high levels of amines in anaerobic conditions, which you may remember if you've ever kept a fish tank. We actually monitor those in fish tanks and things like that. So the Winkler method is a way for us to actually detect the amount or tell the amount of dissolved oxygen present in a water sample. The samples need to be collected, and we need to make sure there's no space that is left in contamination for air, because if you think about it, we want to know how much oxygen is in the water that we collected. If it has air on top of it when that collecting is done, that air can contain oxygen, which will then dissolve into the bottle, so it contaminates our results. There's usually special dissolved oxygen bottles that we use for the collection of this to ensure that that doesn't happen, and the collection it happens under the surface of the water. Once we do that, the sample is fixed by adding manganese sulfate and an alkaline iodine reagent, so in the presence of the base. What that does is the manganese and the hydroxides react with the oxygen to form MnO2, okay, and water. So this is a brown precipitate, okay, the manganese 2 oxide. So that indicates the presence of oxygen, because that won't happen if we don't react with oxygen. So then what happens there is the MnO2 is treated with sulfuric acid and iodide, and what happens is another redox reaction where we end up with Mn2 plus being formed again. So Mn is in a +4 state on the left hand side and +2 on the right hand side. Iodide is -1 and I2, so we have our oxidation and reduction occurring at the same time. Once that happens, the solution now, the higher the I2 concentration that we have here, the higher the amount of dissolved oxygen. And now that we have iodine, we have one of those common oxidizing agents that we said that we could titrate with. So now we have free iodine that can react with a thiosulfate anion, forming S4O6 2- and 2 iodide, which means with this one, if you remember the titration that we did in class with wine, we titrated until it turned blue-black, which meant that we had free iodine left over. This one would actually start that dark blue-black color and go to a colorless at the end point. But you can see from this that the initial amount of manganese that we put in, we can follow the reaction all the way through, and we can see that the amount of iodide, okay, can be linked to the amount of oxygen through stoichiometry. We'll do some practice questions on this because it has multiple equations, but really what you need to do, remember, is that we follow the oxygen and the iodide. So if we have a look here, we're using 1 oxygen up here, and then that process produces 2 MnO2, which would give this one 2O2 here, and we go through and we follow the overall iodine pattern. So how much iodine we get relates back to the amount of oxygen overall. So we will have a look at this in more detail, but remember, you must know these equations and understand the process, because this is one of the prescribed practicals that we need to know about for our course.